Hi class, Mr. Roth here. Today's lesson is on sickle cell anemia. My guess is that you've probably heard of this disorder before, but if I asked you what are the symptoms, what causes it, my guess is that you probably wouldn't have an answer. And so today my goal is to train you how to respond if somebody asks you, what is sickle cell anemia and what causes it? First thing I want you to notice, though, is the image here. You'll notice the red color of these cells, and that might indicate to you that these are red blood cells. And just a little refresher, if you don't know this, red blood cells, really their primary function is to deliver oxygen to the different tissues of the body. And it turns out that a sickle-shaped red blood cell can't do that quite as effectively. And so what this results in is a number of symptoms. Probably the worst one would be the shortness of breath. And this should make sense to you based on what I told you just a second ago, and that's that a sickle-shaped cell cannot bind and carry oxygen quite as effectively, and so the person will experience this shortness of breath. And another word for that actually is anemia. Simply means low oxygen. Okay. What I want to point to here is a sequence of nucleotides, and this is what we call a gene. It's a segment of DNA, and you might notice that the genes are exactly the same, except for one spot. You'll notice right here that there is a mutation, and so this is what we would say is the normal sequence. This is in a healthy individual, and we compare that to this bottom sequence, which is the sequence you find in somebody with sickle cell anemia. Now what I want to point out, this isn't just any random sequence. This is the gene that codes for a particular protein called hemoglobin. This protein hemoglobin, as we described before, is the protein responsible for binding that oxygen and then carrying it around the body. And so it's a very, very important protein. If it's not functioning correctly, you're going to experience that shortness of breath or that anemia. So this leads us to our main question, which is how can just one single mutation in the hemoglobin gene cause sickle cell anemia? It seems like one letter wouldn't make a difference. So let's refresh you a little bit on, actually I have one more picture for you to demonstrate how hemoglobin works and what it looks like. This is hemoglobin, okay, it's this purple protein. And it can load and unload oxygen. If you ever wondered why iron is an important mineral in your diet, it's because iron is part of the hemoglobin protein, and it is actually closely associated with the pockets here that bind the oxygen molecules. And so this protein, very, very important for loading and unloading oxygen, shuffling it around your blood. If you didn't have it, you would die. You wouldn't be able to live because oxygen, as we know before, is necessary in cellular respiration. Okay, so to really understand how one nucleotide change could actually cause a disorder, we first have to refresh ourselves on the central dogma, which is that idea that the sequence of nucleotides here in the DNA is responsible for the sequence in the RNA, which is responsible for the shape and the function of the protein that is made from it. And so in our case, we're studying the gene for hemoglobin and the final product, which is the hemoglobin protein. If we change a letter here, we're going to get a misshaped hemoglobin, and we're going to get some problems with the cell. So where is all of this occurring, and what is it called? Let's look over here at the cell. Here's our nucleus of the cell. This first step, transcription, is simply the process by which we go from our DNA double helix to our single-stranded mRNA. We call that transcription. It's all happening within the nucleus, and once that mRNA is made, we call it a messenger RNA, which means it can leave the nucleus, so it snakes out right here, out of one of the pores of the nucleus, comes out into the cytoplasm, finds a ribosome, which is this round structure, and it kind of feeds through the ribosome. And as it's feeding through, it's being read, it's being interpreted. And these tRNAs, you'll notice that they have a complementary 1, 2, 3. They've got a complementary sequence here of nucleotides that matches. It has to match based on the complementary base pairing rules. And then on the top, it's delivering over an amino acid, which is the building blocks. It's what we're going to make this final protein out of. Now, the order of these amino acids, you can see there's a chain coming out of the top of the ribosome. That chain and the order there corresponds to the order that is coded for in the mRNA. And so consequently, if we change the order, maybe even just one nucleotide in the mRNA, we're going to get a different protein. It's going to be slightly different shaped, which is going to cause some issues, most likely. So again, here's a view of the same exact thing. We are again looking at a ribosome. We're just a little bit more zoomed in. Okay, so it's the same image. And this image has a little more detail, specifically the lettering. And I want to draw your attention to the fact that we have matching here. We have complementary base pairing. We have UUU matching up with AAA. Again, since we're in RNA land, there are no T's. There's only U's. U's are in place of T's, right? But what I'd like to draw your attention to specifically is this coding here. We've actually, over time, cracked the code of biology, and there is a specific code. For example, in the codon, if your codon of RNA says AAA, we can use this chart to tell us what amino acid will go in place there because of the AAA. So let's see if the chart over here corresponds with what the diagram is saying. So to interpret, we got to look at the first base in the codon, which we know is A right here. So there's A. That brings us into this row. Now what column we're in is determined by the second base, which again is A. So that's here. So now we're in this box. We've got it narrowed down to this box. Lastly, which row in the box? The third base determines that. You see A. There's A. And that brings us right here. AAA corresponds to the amino acid lysine. Okay, does that correspond to our diagram? It does indeed. Here is our amino acid lysine, and that will be the next amino acid to be attached to this chain. And so the process continues forward, and coming out in this direction is a large chain of amino acids. Once the process is all said and done, it chops it off, the chain folds up, and we have our final product. In our case, we are talking about hemoglobin. So that would be the final product that's coming out here. Okay, so how then, again, we still have that question, how could one letter change the shape and function of our final protein? Well, let's follow the logic through. If we change the letter here, we get a change in the RNA sequence. If we get a change in the RNA sequence, we get a change in the sequence of amino acids. You'll notice that here, this is the standard pro-glu-glu. In a mutated hemoglobin gene, the final amino acid sequence is pro-val-glu. That's a difference, and that difference is very, very significant, in fact. Having that valine there in that middle position causes the hemoglobin product to clump together. You'll notice that in the normal hemoglobin, it stays by itself. Here we've got 1, 2, 3, 4, 5 hemoglobins all clumped together. They stick together. Now the reason that they're sticking together again is because we've changed their structure. We've made them more sticky by adding the valine. This is a mutation. It's not supposed to be this way. And so what effect does that have on the shape of the cell? Well, that results in the cell having a different structure. And one of the problems is if you have a bunch of sickle cell or sickle-shaped cells running through your bloodstream, they tend to clump up. They tend to get stuck in those very small capillaries in the body, and that reduces blood flow. And one of the causes of reduced blood flow, again, is anemia, shortness of breath, low oxygen levels. That combined with the fact that oxygen is not able to bind quite as well, that altogether is contributing to these symptoms of sickle cell anemia. Okay, so just as a review, again, it's our DNA coding for hemoglobin, codes for RNA, which gets translated into our final protein, which is our hemoglobin protein. Another review here, just to remind you, if we do have that clumped hemoglobin, which is the mutated version, it results in us having a sickle-shaped cell. And then for your normal hemoglobin, we have a very nicely shaped blood cell, which is functioning at its full capacity. So what I'd like to show you now is a quick video that's going to demonstrate to you how oxygen binds to that hemoglobin.
4 molecules of heme, an iron-containing pigment, which binds oxygen loosely and reversibly. Hemoglobin that is fully saturated with oxygen is bright red and is called oxyhemoglobin. On the other hand, hemoglobin that is not saturated with oxygen is purplish blue in color and is called deoxyhemoglobin. It is...
All right, class, there you have it. I think you now hopefully understand the role of hemoglobin and also the way that a mutation can affect its function. We'll see you tomorrow.